All right, welcome back. So this little mini lesson is we're not quite getting to DNA replication yet. I got to kind of set things up a little bit, talk about some of the machinery and the directionality of how these enzymes are going to work. So this is my introduction to DNA replication, and then the next video will be actually getting into the process of DNA replication. So before we get into the machinery, what I call the DNA replication machinery, which is really just a whole bunch of enzymes, I wanted to talk about how they determined how DNA replicated. So the big picture word, if you will, the vocabulary word that describes DNA replication is called semi-conservative replication. There were quite a few hypotheses as the knowledge about DNA was growing. How does this double helix that is made up of all of these repeating nucleotides, how does it go from one DNA molecule to two identical copies? So I'm going to go and skip over the answer, the actual way, that's hypothesis one, and kind of talk about some of the other ways that they thought DNA replicated, and we'll finish up with the actual real way that DNA does replicate. So this first hypothesis was, or the second one in this picture, but the first one we're going to talk about is called conservative replication. And so the idea is, and it's just kind of the light blue ribbons are representing our parental DNA. So here's your DNA. And in the conservative hypothesis, the idea is it splits apart. Each half serves as the template for the daughter strands, which are the dark blue, but then they separate from each other. The dark blues go together and the parental strands go back together. And so here, these would be identical, and this is 100% new. Okay. And in the second generation, the parent strand, this parent strand would still do the same thing to make its new strand, and then this would split to create that. Now, there's a reason why that doesn't work is because what's called perpetuation of mutations, which I kind of have the picture over here on the other side of the screen, which we'll get to in a little bit, but they knew based on genetic analysis that mutations are perpetuated through generations in very specific ways. And this conservative replication did not explain how mutations were perpetuated from generation to generation. It just, the evidence did not match that hypothesis. So that hypothesis was invalid. It was not a good explanation of DNA replication. Okay, so poo-poo on that idea. The next one that we'll talk about is called dispersive replication. The idea being that your parental strand kind of unzips, breaks apart, copies, and then randomly comes back together in all of these weird patterns so that you have new and olds kind of randomly distributed in your parent strands and your daughter strands. And again, that was not supported by the evidence of tracking these mutations through generations. So nope, that hypothesis was not supported. So to discuss the hypothesis that was supported, and this one's called semi-conservative, or conservative, the parent strand was conserved. It was not given to any of the daughter strands. Well, in the real way, in the actual way that DNA replicates, the semi-conservative model, is you have your parent strand, it splits, it serves as the template for the daughter strands, right? So we're putting new A's, T's, C's, and G's in place. But instead of separating the new strand out like we saw in the conservative replication, the parent, right? So the parent and the new daughter strand, the light blue and the dark blue, stay stuck together. And so your daughter strands are one half the original parent and one half the newly formed complementary strand. So then each of those in the second generation would serve to be the parent strand, and then the newly formed daughter strand. So if they wanted to do a better job at this, they should have made one of these a different color, showing that it's actually the third copying of that light blue parent strand. And this is how the evidence, this is what the evidence supported as far as a semi-conservative and this propagation of mutations, because if you have a mutation at this point here, it will be propagated in 1/4 of your offspring. And so that's kind of what they saw following kind of Mendel's genetics that we covered in chapter 11, because one half will be normal, where the mutation was, it will lead to a mutation on both strands. And so now any daughter cells of that particular mutated strand will maintain and propagate that mutation, where all of the other daughter strands will be normal. And so that type of evidence helped to support this semi-conservative hypothesis of DNA replication. Okay, so in a molecule of DNA, whether you're talking about a bacteria, which is a circular piece of DNA, or a linear piece of DNA, before we can actually start the whole replication process, we have to open up that DNA. Sometimes it's called unzipping, is breaking those hydrogen bonds and opening that up a little bit so all of the enzymes and replication machinery can get down in there. And this is what we call a replication bubble, or origin of replication. So the origin of replication generates a replication bubble, and that bubble will keep getting bigger and bigger and bigger until bubbles meet. They'll kind of go and kind of towards each other, and they'll meet, and then we have two identical daughter strands. Our DNA, like this, is eukaryotic DNA, linear, right? We have 46 individual pieces of DNA, which are our chromosomes. Bacteria are circular. They only have one circular chromosome. So when they just make one replication, they have one replication, origin of replication, and one bubble, and then the bubble just grows and grows and grows and grows and grows until it meets itself back in the middle, and then voila, you get two identical daughter chromosomes, just like you do in eukaryotes. But in bacteria, it's a circle. In eukaryotes, it is a line, a linear piece of DNA. Within that replication bubble, you can kind of see all these little pink arrows that are pointing in these micrographs. The arrows are pointing to what we call the replication fork. So the replication fork is the point at which, right, your double-stranded DNA is being split and getting ready to be the template for new DNA being made as the daughter strands. So this area right here is what we call the replication fork. Okay. So let's take a look at what I call the molecular machinery. These are two different pictures of the same idea. I think the bottom one might be from your book and the top one might be from a different book. I don't know exactly where the pictures came from, but I was just wanting to give you two different views of the same collection of enzymes, which I call the replication machinery. So the first enzyme that goes at this origin of replication and kind of helps to open up that bubble is what's called helicase. So it's this little brown wedge here, or it's this green dumbbell-shaped enzyme here. And we know it's an enzyme because it's got the ASE ending, helicase. So what this does is it finds that replication bubble and goes into that replication fork and breaks the hydrogen bonds. So helicase breaks the hydrogen bonds, separating the two parental strands. So they're being separated and pulled apart, breaking those hydrogen bonds. But the whole nature of hydrogen bonds is they're attracted to each other. They're polar attractions, right? So the partial positives and partial negatives on those bases, they want to come back together. So we have to have what are called single-stranded binding proteins. They're kind of these gray ovals here and the green circles here on the bottom picture. Single-stranded binding proteins kind of come in and block the newly separated DNA strand from coming back together, because we don't want it to come right back together. That defeats the purpose of trying to replicate it. So helicase opens the double helix, breaks those hydrogen bonds. Single-stranded binding proteins keep those newly separated strands separate from each other. And then as you are opening and unwinding the DNA, it's a helix to begin with, but if you're kind of opening it up like this, it might supercoil and potentially damage some of the DNA downstream. So here we have this enzyme called topoisomerase, blue in the top picture, purple down here in the bottom picture. What it does is it kind of works a little bit upstream to the replication fork. It kind of breaks some of the bonds between the nucleotides and unwinds some of that tension, and then reseals it back up and then moves down the line a little bit. So as that replication fork is opening, we're not causing damage to the downstream DNA. So the topoisomerase, sometimes called gyrase, but I think your book calls it topoisomerase. What it does is it helps to relieve the tension on the upstream unseparated parts of the DNA. So you can remember that. And then lastly, the super important enzyme here is called a primase. excuse me, sometimes it's called the RNA primase or just primase. This is a really, really, really important part of the beginning or the initiation of DNA replication. And I'm going to introduce to you some rules of DNA replication. One is DNA polymerase, running out of room, is the enzyme. So that's not shown in this picture, but it is the big enzyme. DNA polymerase can only add bases to the 3' end. There's that 3' again. I told you it's going to be important, and my 3 looks horrible. So let's fix that. So the DNA polymerase, the primary enzyme of replication, can only add bases to the 3' end. The second rule is DNA polymerase, we're just going to call it DNA poly for short, cannot start on a single DNA strand. This is where the primer comes in. So let's say I'm gonna draw a DNA polymerase that's this big purple blob. So if I were DNA polymerase, I would not be able to land on that single strand of DNA. It just doesn't work. That's rule number 2. But what I do need is I can land on a short little double strand. So that's what the little RNA primer, that little red bit here. So because of that, RNA primase, which can come in and land on a single strand of DNA, I can then come in as my DNA polymerase and land on that short little piece of double-stranded, well, half DNA, half RNA. And then it will allow me to do my first rule, which is adding new bases to the 3' end, right? So there's my 3' end. My DNA polymerase, which is my purple blob right here, can then start adding bases to the 3' end. So the primase sets down almost like a landing pad for the DNA polymerase to land on, and it provides that unbound 3' end for the DNA polymerase to start laying down the new bases that are complementary to the parent strand. So we're going to see these 2 rules as we continue to go through the process of DNA replication. So DNA polymerase can only add to the 3' end, and DNA polymerase cannot start on a single DNA strand. It has to have some kind of docking pad, and that's what the RNA primer is there for. So here's a picture to identify that second rule. DNA polymerase can only add new nucleotides to the 3' end. So again, when we are taking a look at those carbons, and actually there's oxygen here. This is a carbon, 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 and then our phosphate group, right? So this is 1', 2', 3', 4', 5'. Because DNA polymerase is an enzyme, it has a very special active site, right? We know that about enzymes. And its active site only allows for new nucleotides to be added to the exposed 3' end. So here, this would be the 3' end of this nucleotide. Okay? And so coming into it, this enzyme is going to bring in the 5', because that's where the phosphate group is hooked to. So the 5' of this newly incorporated nucleotide binds to the 3' end of the existing nucleotide, and then we know its partner because T goes with A. We would not be able to add to this end. No, no, no. DNA polymerase does not work that way. That was rule number 2. 1, DNA polymerase can only add to the 3' end. So we can only add bases to these exposed 3' ends. All right, so that's kind of like our introduction and kind of setting the stage, and we will move on into DNA polymerase on the next video. Okay, bye.